PCR has been used in various fields in the molecular biology, uh, like genetic fingerprinting. So it's compare the person with a given sample, such as blood samples are found in the crime scene, and that sample may contain a very small amount of DNA. But the application of PCR comes at this part to amplify the small amount of DNA from these blood samples or the segments of saliva here. So it is used for the genetic fingerprinting, printing, and the amplified DNA fragment then further analyzed by gel electrophoresis to the comparison of the DNA band of the suspect and sample found in the crime scene. Also, it has been used for the detection of hereditary diseases. So that again, PCR, the main purpose is to amplify a small amount of DNA collected from the patient, and that can be used for DNA sequencing to determine the mutation inside the gene. Cloning, as we have seen in our previous lectures, that is an integral part of PCR has been an integral part of the cloning. So you need to be used for the cloning for the DNA fragment, or sometimes amplifying the vector as well, the fraction of vector, fragments of vector. Analysis of ancient DNA, like the genetic fingerprint or the hereditary diseases. So the small amount of DNA can be used to amplify again and compare with the recent time of the evolutional analysis kind of thing. Quantitative PCR or the qPCR is a method that is similar to PCR, but there are some differences between the PCR and the qPCR. So it enables to determine the exact amount of DNA, amplified DNA into the samples, and it has been really used to quantify the transcript, the RNA transcript, that is the messenger RNA, to measure the gene expression level. So in this table, we can see that there are some differences between the PCR and the qPCR. So the PCR is more of a qualitative technique, whereas the qPCR is a quantitative technique. And the product has been detected by the agarose gel electrophoresis. On the other hand, the qPCR, the product can be detected in each amplification cycle. So as the cycle is running and every cycle is repeating, and you can get a detection from the amplification of that cycle. And the data is actually collected at the end of the reaction in PCR, whereas in qPCR, the data is collected in every phase of the reaction or every cycle. And this is a very high resolution, the qPCR, and it uses the fluorescent dye to detect the product, whether in PCR, it's normally used the ethidium bromide detection method to get the PCR. So sometimes the PCR is more time consuming than the qPCR. So these are the differences between the PCR and the qPCR. So for the qPCR, we generally used a cDNA. cDNA is a complementary DNA, so complementary to the messenger RNA. So from the messenger RNA, DNA has been prepared, which is also known as the first strand DNA. So as it is exactly complementary to the messenger RNA, that's why it is called a cDNA. And the process which has been used to get a cDNA from the RNA is a reverse transcriptase, which is an enzyme that is actually using the RNA as a template to make a DNA. And from that cDNA, using the TNTPs, that is the bases and the enzyme, normally you can, reverse transcriptase, we have already used the enzyme, and then we can make a first stand in one single experiment to make the first strand, and from using that first strand, we can make thousands of copies of those fragment of cDNA. And in that way, it is actually giving in a qPCR machine that those bases are attached with the fluorescence particle. So they are actually giving the signal to the equipment, and we can get actually detected by those signals. Then we can also prepare a DNA library. So this is a collection of the DNA. Normally is a genomic DNA, and there is a high probability that's finding any particular piece of source DNA into that collections. And the DNA library normally prepared either from the genomic DNA or from the cDNA. So the genomic DNA is all of the genes involved in any organism has been used to make the genomic DNA. And for the cDNA is actually those genes that are expressed into the messenger RNA. And from that messenger RNA, it has been making a cDNA, the complementary DNA from those messenger RNA, and making the library. The use of this is obviously it's explore the genome of an organism and learning the more about the genomic structure and the functions, and also the cloning of a segment of DNA. I have included a video of how you prepare the genomic DNA on the cDNA library. It's more of a same of a molecular cloning. So in both cases, you get the genomic DNA extract. In cases for the genomic DNA, you extracted all the genomic DNA and then you fragment them. And after that, you ligate them into the vector, and that plasmid has been transformed into the bacteria, and the bacteria has been going into the plate, and all of the bacteria has been collected, and that's your genomic DNA library. On the other hand, the cDNA, all of the messenger RNA has been extracted, and then using that messenger RNA, you make the cDNA, fragment that one, and then ligate it to the vector, and the other steps are the same. So that's where we normally make the DNA library. And this is just a common process of making a genomic library. So you extract all the genomic DNA, and from that genomic DNA, you actually make a small fragments of genomic DNA, either using by restriction enzymes, and then those DNA fragment has been cloned into the vector, and using a ligase, and then transformed into the bacteria. And the bacteria, all the bacteria glued into the plate has been collected and used that bacteria as a genomic DNA library. So next generation sequencing, this is just a bit of touch to the next generation sequencing for you guys. And there are a number of platforms has been used for the next generation sequencing. I have included one video for the next generation sequencing of the Illumina. They use a sequencing by synthesis. It's very interesting. Just have a look at the video. I hope that you will understand, and definitely we will discuss, we can discuss into the discussion classes if you have any question, but this is just general information for your right at this moment. I don't think that you really require this next generation sequencing right at this moment, but just get an idea about how the next generation sequencing has been performed. Thank you.